Okay, so today we're going to be discussing polymerase chain reaction, what it is, the materials that we need to complete the process, and the ways that we can utilize this in everyday life. So the first question that we ask ourselves is, what is polymerase chain reaction? So polymerase chain reaction is also known as PCR. That's how I'll be referring to it for most of this video. So PCR is a technique that is used to make copies of a specific strand of DNA. It can be a very small strand, a specific gene, any length of DNA, and PCR can generate thousands to even million copies of an exact piece of DNA. PCR was invented by a man named Kary Mullis in 1983, and he won the Nobel Prize for it in 1993. It has been disputed that he should be the sole winner of the Nobel Prize for this award because without work that had been done previously on DNA research, he wouldn't have actually been able to come up with this information. But ultimately, he was given the Nobel Prize because he had the idea to bracket the desired DNA using a pair of primers and amplify that particular strain using DNA polymerase, which is what I will get into later. He was also kind of a black sheep of the biological world. He is known for coming up with this technique while tripping on acid. He also didn't believe in the connection between HIV and AIDS, and he thought that climate change was a hoax. So he was very much a black sheep of the world, but he did win this Nobel Prize. So the materials that we need to get started with PCR, we are going to need the actual DNA template, the sample that we're actually amplifying. And then the next thing that you're going to need is your DNA polymerase. The polymerase that is used today mostly in PCR is called Taq polymerase. It is a thermostable DNA polymerase. It is named after a bacterium Thermus aquaticus, which was isolated in 1976. Originally, DNA polymerase was used from E. coli in PCR, but it couldn't withstand the high temperatures and it would have to be redone every single process, so it just made it take too long. So when the Taq polymerase was isolated, they started to use that in this process. You're also going to need primers, which are short strands of DNA that adhere to the DNA strand. They provide a starting place for the replication, so that just gets the whole system started to go. They attach to each single-stranded piece of DNA. I'll talk more about that later. You're also going to need your nucleotides, so you need the building blocks, so you need to have something to add on to the specific strain of DNA that you're trying to replicate. And you also need a buffer, which is the solution that you do this whole process in, and the pH of this buffer is normally between 8 and 9.5. So there are three main steps that are a part of PCR. The first step is denaturing, the second step is annealing, and the third step is extending. I will go into detail on all three of these steps in the next following slides. So the first step of PCR is to denature. The main purpose of this step is to take your double-stranded piece of DNA and you separate it into two single strands. So this is done by breaking the hydrogen bonds in between the two strands of DNA. And hydrogen bonds are pretty strong, so you have to heat up your reaction mixture pretty hot to actually get the strands to denature. It stays up about 95 degrees Celsius. And your first initial denaturation is going to take longer than the ones after that. Just so you get the initial breakage of the hydrogen bonds, it'll take about 2 to 3 minutes, but after that, during the cycles, it should just take a couple seconds to maybe a minute every single time that you do it. So after your denature step, you have your anneal step. So this is the second step, and this is where your primers bind to the newly single-stranded DNA. So for this to happen, we take the reaction mixture temperature from about 95 degrees Celsius, where we were at the denaturing step, down to 45 to 60 degrees Celsius. So the temperature should be around 5 degrees Celsius lower than the melting point of the primers that you're using to bind to your new DNA. This step will only last about 30 seconds to a minute. It's just enough time for the primers to bind to the complementary strand of DNA. Your last step after annealing is going to be your extending step. So this is where the DNA polymerase, the Taq polymerase we talked about earlier, will extend all the way down both strands of DNA to create a new complementary strand of DNA. So the reaction mixture is heated back up because the annealing step, remember, it was at 60 degrees to 45 degrees Celsius. This step needs to be heated back up to 72 degrees Celsius. And this step normally takes a minute, depending on the size of your DNA. So if you have a particularly large strand of DNA that you're trying to amplify and replicate, say it's longer than 2 kilobases, it'll take an additional couple minutes depending on the length. And over here on the right side of the slide, you can just see how many actual copies of DNA you can get off of just a few cycles of PCR. This is just a breakdown, a quick little picture of the whole process together, the denaturation step, the annealing step, and the extension step, and this happens several times throughout a reaction. So how do we view the results of PCR? So we view the results by a method called gel electrophoresis, which was talked about in other videos. So gel electrophoresis separates the DNA fragments that you have according to size, and it's pulled through your gel by an electric current. So the smaller the piece of DNA, the farther that it's going to travel through your agar gel, and it'll give you a marker, and it'll line it up to the base pair on the left side of your agar, which is your control. So there are several different types of PCR. I just have a few listed here. There is definitely more than this, but I'm going to go into quick detail on what each of these are. So real-time PCR monitors the amplification of targeted DNA during PCR. So the reason this is so relevant right now is it's a lot less time-consuming than regular PCR, and you can get your results after each single run. So you can actively view the results as the PCR is happening, and you don't have to wait till the whole end of the cycle is done. Reverse transcriptase PCR, which is RT-PCR, which is used by RNA. So we use RNA and we convert it into complementary DNA or cDNA by reverse transcriptase, and then we use PCR to amplify that copy DNA. And the reason that we changed the RNA into copy DNA is because RNA is quite unstable and it can degrade pretty easily. So that's why reverse transcriptase PCR is so important, because it gives us a chance to study the RNA at a more complex level after it has been turned into cDNA. There's also something called nested PCR, which is just a way to get a more accurate run of your PCR and not have as many complications or unexpected products. So the way that this is accomplished is it uses 2 sets of primers instead of the 1 set of primers, and it runs 2 successive runs of polymerase. So essentially the way I like to call it, it's PCR inside of PCR. So your first primers will attach farther downstream DNA, and the second pair of primers will come in and attach inside that original pair of primers just to get you a more specific, uncontaminated product. And multiplex PCR is a quite new, but it is very relevant today. So it refers to the ability to be able to run multiple pieces of PCR at the same time. So if you have multiple strands of DNA that you're trying to amplify, say you're working at a forensics crime scene and you're wanting to compare the suspect's blood with the blood found at the crime scene and another suspect's blood, you can run all 3 of those samples at one time, so it saves a lot of time and aspects like that. So what all can PCR be used for? Like I said a minute ago, it can be used in criminal cases, but it can also be used in the early stages of DNA sequencing. It can determine the order of bases and the length of DNA. Right here we have a picture of the multiplex PCR, which I was just talking about a minute ago, and how that can go through the PCR, how you can run several different components at a time, and you can compare it on the agarose gel. PCR can also be used for the detection of some diseases, including genetic and autoimmune diseases, and it helps identify a lot of the causes for diseases that we thought would be idiopathic. And idiopathic means something that we thought that didn't have a reason for why it happened. So this is explaining a lot of gene mutations, some things that we wouldn't used to be able to explain, but today we have a reason for them because of PCR. There's also been methods developed that are capable of identifying contaminations in food, like whenever we have huge recalls on lettuce or spinach because of E. coli and salmonella, this multiplex PCR system can get the results in about a day versus the normal conventional culture method would take about 5 to 6 days. So it's a really big time saver in that aspect. PCR can also help in the detection of infectious agents. So it is specifically very helpful in detecting HIV because HIV takes a long time to show up in traditional tests because normally you have to wait for the antibodies to be produced in your body. But if you're using PCR, you can get results almost immediately after your infection occurs because of how quick it can do. And even if you do get a positive HIV test, PCR can tell us how much of the virus you actually have inside of your body. This is called a viral load test. So it can actually show you the specific levels of a specific virus that you have in your blood, and it can help you figure out if any treatments are working. PCR can also be used to identify the causative agent of anthrax. This is also super important. It's another time-sensitive thing. It can detect the anthrax in any specimen. It would normally take over 24 hours, but with the use of PCR, you can get results in an hour or two, which is very, very important, so you can start treatment as soon as possible. And lastly, PCR and cancer diagnosis. So PCR has helped a lot with leukemia patients. Like we said earlier, how the viral load test can show us how much of a virus is in someone's blood. Well, it's been used a lot in leukemia treatments to see if the treatment is actually working. So if you run PCR on someone 1 month and then you run it again 2 or 3 months later after they've had this treatment, you can see how much of the virus is in the body and if that specific treatment is working for them or if it's not. And so you can get almost individually tailored treatments like which is showed here. So you run the PCR and you can decide whether this medication will work or whether this therapy will work. So it's a very, very, very important tool that we can use in diagnosing things right now.